For this lunch and learn, we're going to hear from Dr. Paul Grossfeld, and so I'm going to just read his little bio real quick. We all know him, right? But we're going to still read his bio, and there are some people who may not be familiar. So Dr. Paul Grossfeld is a board-certified pediatric cardiologist on the faculty at UCSD School of Medicine. In addition to treating pediatric patients with heart disease at Rady Children's Hospital of San Diego, he runs a research lab at UCSD dedicated to discovering genetic causes of congenital heart disease. Dr. Grossfeld has served as a chief medical advisor for the 11th Research and Resource Group for over 20 years and has published over 20 papers on Jacobsen syndrome. He also served as the director of Pediatric Cardiology Fellowship Program at Rady's for 12 years, during which time he oversaw the training of over 20 future pediatric cardiologists until he stepped down in 2020 to devote more time to his research. He's also served as a cardiology consultant for the U.S. men's and women's Olympic volleyball teams for the past 12 years. Dr. Grossfeld and his wife and son live in San Diego, enjoy international travel, and have led over 10 annual medical missions to Cambodia to perform life-saving heart surgeries on children there. And Dr. Grossfeld has a passion for music, photography, and most sports. He's coached several of his son's basketball and baseball teams over the years. Some of his current passions include snorkeling in La Jolla Cove and golf, tennis, and learning classical guitar. We'll pass it over to you.
Thank you. Thanks, everybody. It's actually really powerful. I look out, I see the Washington Monument and the Pentagon. It's like such a unique setting in so many ways. Curtis actually shared a story with me today that I completely forgot about. I think you were telling the truth. And that was, about 20 years ago, 21 years ago, whatever, there was actually a lot of instability in this group to the point where there was actually a really good chance that this group was just going to dissolve. And that's when Lindsay, I think, expressed an interest to step up to take the role of president. And Curtis reminded me that I guess I said at one point to Lindsay, if you don't become president, I'm not going to have anything to do with this group anymore. I don't remember saying that. I can't believe you didn't call my bluff on that. Anyway, thank you. And truly, I think we really owe a huge amount of thanks to Lindsay and Jessica and Rich Siritz. I'm not sure if he's here today. And where's Beth? It just takes so many people that work behind the scenes predominantly, not just for the few days here at the conference, to make this organization so successful. And I'm personally incredibly proud to be part of this. I think it's just a model for how a support group for a rare genetic disorder should work. So thank you guys for all that. So how many of you saw the movie Bohemian Rhapsody? Just out of curiosity. Oh, you guys know. No, I'm not going to do this, but at the Germany conference, I pretended to be Freddie Mercury. Hey, you want to come up, Craig, and lead that? Come on, you've got a voice. All right, so I think that's all my humor for the day. I will start off telling you about what my original goal has been. And, I look over there and there's a young lady who is really the whole reason why I've been doing this. She's an example, a case in point. Maybe you could say she's doing so well despite everything I've done. So anyway, so what I'm going to tell you today is actually a pretty structured talk with a lot of details. Don't worry about understanding every detail, although I am going to ask Jessica every so often to make sure that she gets it, and if not, you need to stop me. Okay. So really kind of from the get-go, what for the cardiac perspective and really for the other issues as well, what can we learn from Jacobsen syndrome about congenital heart disease and the genetics that could hopefully apply to the general population? And just to review what I said the other day, about 1%, almost 1% of children that are born have some kind of a congenital heart defect. And the vast majority of those cases, even though we do a great job treating these patients, but in the vast majority of cases, we don't really understand anything about the underlying cause or the mechanisms. So you have to think that as we learn more about the specific causes and mechanisms, that we'll come up with better therapies and maybe even preventive strategies. So let's see. This projects all right. So what I want to tell you is the specific heart defect that I'm most interested in called hypoplastic left heart syndrome. And this is, you know, if you go to the internet or go to a cardiology textbook, this is the typical type of diagram you'll see. So on the, is that your left? Yeah, on your left is just a cartoon of a typical heart. So you see the four chambers: left atrium, left ventricle, right atrium, right ventricle. You see the main artery supplying blood to the body called the aorta, and then you see the main artery supplying blood to the lungs called the pulmonary artery. And red because that's oxygenated blood going out to the body, and blue because that's desaturated or unoxygenated blood that has to go to the lungs to then come back to the left side of the heart. So in hypoplastic left heart syndrome, at least is depicted here, the left ventricle is too small. In other words, that chamber volume is too small to be able to pump enough blood out to the body. And so these days, most babies actually with hypoplastic left heart syndrome are actually diagnosed prior to birth, prenatally. But 22 years ago, that was not the case. That was actually much more common, that babies would present after birth, usually in some kind of dire situation. So as you can see here, not only is the chamber volume itself very small, but the valves on this side of the heart compared to on the normal heart, this is the mitral valve, which tends to be very, very small, sometimes completely blocked off. So we call that atresia of the valve. And similarly, the aortic valve can be either very small, what we call stenosis, or completely blocked off, atresia, okay? And as you see here, the way this is drawn, it suggests that maybe in this case, it may be due to sort of this normal heart tissue here that lines the chamber kind of growing inward, okay? So that's one possible explanation, and I'll come back to that. And by the way, given the fact that it is lunchtime, I just want to forewarn you, I will show you a few pictures of actual human hearts. If you want to close your eyes during that, that's fine. I don't think it's too gross. I think actually they're beautiful. But anyway, just be a little bit forewarned for that. I'll try to let you know ahead of time. Here we go. Okay, close your eyes, everybody. That's not too gross, is it? All right. But actually what we call hypoplastic left heart syndrome is actually a group of heart defects. The common denominator, so these are the four different anatomic variants, and they vary in terms of whether those two valves on the left side are actually just small or actually completely blocked off. So if you look at the lower left picture, this is actually the version that seems to happen most commonly in Jacobsen syndrome, in which the valves are actually blocked off. So we call that mitral and aortic valve atresia. And going along with that, you can see this little tiny slit. That is the chamber volume that's was meant to pump blood out to the body. Obviously, that's not going to work. The other typical way that this particular version is described is a slit-like left ventricle. And so I think you can appreciate that's really what it is. And again, the mitral and the aortic valve are totally blocked off. Kind of at the opposite end of the anatomic spectrum is what I'm showing you on the upper left corner. So once again, you have a very small chamber volume, so definitely not able to sustain the cardiac output. Here, though, you have actually this scar tissue that lines that, and I'm going to come back to what that is. There's normally a layer of cells that forms the inner lining of the heart called the endocardium, and I'm going to come back to that because we're learning a lot about how that particular layer of cells is not just there for decoration, but actually has, first of all, a very important role in the normal development of the heart, and our evidence, as well as that of others, is beginning to build a case that the endocardium may actually have a critical role in the pathogenesis of hypoplastic left heart syndrome in at least some, maybe all cases. But the other thing that's really fascinating about this heart compared to this heart is look at this wall. This is massively thickened, right? So even though it's called hypoplastic left heart, I can tell you it's amazing in the literature. It's so understudied. Nobody actually acknowledges in the literature that this is not hypoplastic. In fact, my colleague at Rades, who's a pathologist, appropriately calls this hyperplastic. And maybe we should, in this case, call this hyperplastic left heart syndrome. And no one has really delved into that. And so one question about all this is, are these really two similar variations of the same disease process, or are they completely different? And actually, I think as we learn more, even though the common denominator of a very small chamber is the same, I actually think they're two very different diseases, and I'll be speaking to that in future slides. So this is just another example of a young man's explanted heart who underwent a successful heart transplant. And again, you can see this massively thickened ventricular wall. This is the outline of the left ventricle. You see that scar tissue, and if you take a little sample of tissue, like right there where my arrow is, what you see here is just pure heart cells. These are sort of the working cells that actually do the contraction. They're called cardiac myocytes, okay? And I think you could probably appreciate that in this situation, one would have to, again, argue that it is actually an overgrowth or too much cell division that leads to this very massively thickened wall. You with me so far, Jessica?
I'm trying.
All right, just too much cells. Okay, to give you a little bit of perspective about hypoplastic left heart syndrome, it's probably still considered the greatest cause of death in infants born with congenital heart disease. But fortunately, once you get past, if you get past the first year or so, first few years with the 3 surgeries, people actually, as we know, can do very, very well, well into adulthood. In Jacobsen syndrome, I talked about a couple days ago, it's a little bit debatable, but it's certainly one of the most common causes, maybe still the most common cause of death in infants usually that are born with Jacobsen syndrome and hypoplastic left heart syndrome. It is not trivial. I can tell you that almost any time, if you go to my hospital, there are going to be kids in the hospital with hypoplastic left heart syndrome, usually the younger kids having problems. And, you know, just from a very monetary standpoint, it probably costs well in excess of $1 billion a year for healthcare costs directly, not to mention all the other indirect costs of people, family members missing work, emotional impact. It's a big deal. And this is kind of a cartoon showing the most commonly used approach to palliate patients with hypoplastic left heart syndrome. Basically, what we do is ask the right ventricle, shown here, to do the work that this left ventricle is no longer able to do. So, and instead of blood being pumped by the right ventricle to the lungs, the right ventricle is then hooked up to the aorta. So that procedure is called the Norwood procedure. And the blood that was supposed to come back to the right ventricle to go to the lungs is actually directly hooked up to the lung arteries. And that last procedure is actually called the Fontan. So this is a surgical palliation. And as I said, we've come a long ways. And as we know, there are much better outcomes now than how it used to be. But again, as I sort of laid out for you, there are these different versions, and very, very little is known about the underlying mechanisms. There have been some theories. I'm not going to go into that right now. But what's been very limiting is, for a variety of reasons, unlike other diseases, there haven't really been any good animal models to study hypoplastic left heart syndrome, let alone the different forms. And if you look at human heart tissue specimens, we're actually starting to do that. There are some very interesting observations, but you're always stuck with asking whether or not that's some secondary effect after being born, or if that's actually part of the primary disease process that causes it. So for those of you who have been coming to these conferences for the last 22 years or so, I always like to show this picture because I still see this young man. This was taken in July of 1995 when I was in my first month of cardiology training in San Diego. And I was taking care of him for his heart problems, which were kind of a mild form of hypoplastic left heart syndrome. And I mentioned this the other day. This is a picture of, you will never see this almost anymore, but this is his chromosomes, called the karyotype analysis, which Dr. Martina mentioned briefly. And I think you can easily appreciate here how there is a missing chunk of the long arm of chromosome 11. So that's Jacobsen syndrome, and Dr. Jacobsen's original paper was actually in his chart, his paper chart back then. And so one could, you know, you didn't have to be a rocket scientist to speculate that there must be something interesting in that part of the chromosome that presumably was causing the heart defects, and for that matter, the other problems as well. And so again, truly thanks to these conferences. I don't know, Annette, if you remember that first questionnaire that I gave out to all of you guys. And after several years and a lot of long distance phone calls that my boss at the time came down hard on me for, we learned a lot, seriously, just by asking and listening. And that led to the paper that we published in 2004. on 110 cases. And prior to that, I think the largest number of cases published was about 17. So, you know, when you hear that there are only 200 cases or so known in the world, yes and no. There are actually about 250 or so cases that have been published, but in the United States, we think there are probably like 1,000 people if you extrapolate from how many I know that are in San Diego. Okay, in Jacobsen syndrome, as many of you know, just from the show of hands the other day, about half of your children have heart defects. So even if two people have the same exact deletion, one might have hypoplastic left heart syndrome and the other might have a completely normal heart. And I'll touch upon that maybe a little later on. And interestingly, somewhere between 5 and 10% of all kids born with Jacobsen syndrome have hypoplastic left heart syndrome. Are there any others besides Parlin in this room? I don't think so, but that fits about right, 5 or 10%. So if you compare that to the general population, it's like 200 times more frequent than the general population. And probably if you take all comers with hypoplastic left heart syndrome, which is about 1,000 to 2,000 babies born every year in the United States, maybe about 1 to 2% of those are actually also associated with Jacobsen syndrome. Like I mentioned, it's probably one of, if not the most common cause of death in people with Jacobsen syndrome, almost always babies or young children. And as I mentioned, and I'm going to come back to this a little bit later, that particular form that I showed you with that very slit-like left ventricle with the complete blockage of the valves on that side seems to be the most common version in Jacobsen syndrome. And actually, for me as a researcher, it's a great syndrome to study because I don't know of any other syndrome that has as high a frequency. So again, to reiterate what I said before, I'm hoping, and I think I'll show you already, how the progress we're making in some of your children are actually extending to the general population as well. And actually, as borne out here, we could probably spend the rest of the time just hearing everybody's stories, but it turns out that most of them common heart defects that occur in the general population also occur in Jacobsen syndrome. And quite recently, I think we're beginning to learn some insights as to why one person might be born with a normal heart, another with hypoplastic left heart syndrome, and another with total anomalous venous connections. I'll address that a little bit later on. And so this is some technology that came along back around the early 2000s, what people refer to now as an array CGH or comparative genomic hybridization. It is far more sensitive to map exactly where your child's deletion breakpoint is. And from that, we can get a readout to know exactly which genes are affected. And that allows me to help give you guys some guidance about what kinds of things to expect. So even the other talks, without this technology, we wouldn't be able to say with any specific ability how your individual child may or may not be affected. And so based on some very interesting patients, that blue line actually is representing the deletion of that little girl I showed you the other day from Colorado who actually got a heart transplant, but her deletion was much smaller than others. And so that already narrowed it down to this part of the chromosome, which was about half of the original size. And then we came across several very rare patients that, rather than having a terminal deletion to the end, they had these so-called interstitial deletions. Let's say here, also with hypoplastic left heart syndrome. And if you hypothesize that it's the same gene causing hypoplastic left heart syndrome, both in this with the blue and this with the red, then you could hypothesize that that gene must be in this very small region of overlap. And indeed, when we look at that region of overlap, there was one gene in particular that stood out as the very likely cause. And many of you have heard me talk about this, ETS1 or ETS1. And the other day you heard a little bit about something called a transcription factor. And transcription factors are really important genes. They're basically like molecular switches. And as you heard from Dr. Lambert's talk the other day, that's what determines, let's say for a cell, since basically all cells start out with 23,000 genes, right? But each one has to be programmed in a unique way to become a heart cell versus a blood cell versus a lymphocyte versus anything. And so it's the specific transcription factors, these molecular switches then that turn on specific genes that are ultimately expressed to give you that specific type of cellular structure and function, if that makes sense. So ETS1 is a transcription factor, and it had already been shown to have a role in heart development in an organism called the C-squirt. And so I won't go into all the details, but of these 6 or so genes, this one really clearly stood out as being the likely gene for causing maybe even all the heart defects in Jacobsen syndrome. All right, Jessica, how are you doing?
I'm not ETS1. I mean, I
Perfect.
did say that to you yesterday.
Yeah, see, that's progress. All right, you get an A+ so far. Okay, so one of the things in order to understand the actual biology of what this gene is doing is to look in the mouse, in this case in the mouse heart, to ask where is it functioning and when. And so the heart is not just muscle cells contracting. There are these other groups of cells that have different functions, both as the heart is developing and later on for function. And this is just to show you wherever you see this blue, this is where the ETS1 transcription factor is actually being expressed and presumably functioning. And when you look at that, we can actually show that it coincides with 2 different populations of cells that are already known to be critical for the normal development of the heart. One is called the neural crest, and the other is what I alluded to already, this inner lining of heart cells called the endocardium. And just as a little aside, every so often in the Facebook thing, people want to know, why is it that there's so many kids with Jacobsen syndrome that have red hair? Does that sound familiar?
Yes.
Well, neural crest cells, among other things, actually go on to become the cells that make up skin color. And so I'm pretty sure that because of what I'll show you in a moment, the effect of ETS1 on neural crest cell function, that's the gene that's responsible for so many kids having red hair color. Our mice actually that normally have black fur, if we eliminate that same gene, they have white fur. So yeah, see, so that's my argument. A little bit of useless trivia for you. So here, within the neural crest cells, besides other features, structures, including craniofacial features, there is actually a specific subset of neural crest cells called the cardiac neural crest. And this is just a really nice cartoon that sort of depicts that. And it's actually a really incredibly beautiful biological system. So neural crest cells, by definition, originate in the developing spinal cord in the embryo, what we call the neural tube. And as you can see here, a very specific set of these cells, cardiac neural crest cells, actually have to migrate all the way down from where the neural tube is to the developing heart. And they actually go on to form some of the final structures of the heart. And I'm going to show you more information about that in a minute. But just stepping back, knowing that ETS1 is expressed in these 2 populations of cells, the neural crest and the endocardium, but interestingly not cardiac myocytes, then it suggests that it's the loss of ETS1 in either or both of these populations of cells that is responsible for causing heart defects. And so what I'm going to do is spend a little bit of time actually telling you about our earlier studies that are just about to be published, we think, describing the role of ETS1 in the neural crest population of cells. And what you see here on the upper left is actually a cardiac MRI, just like probably some of your kids have had, of a newborn mouse heart. And this, just to give you an idea of the resolution, this little white line here is 1 millimeter. And you can see here, that's a normal left ventricle. That's a normal right ventricle. This red blood vessel here, what's that called, Jessica? It's the aorta. So that's the main blood vessel. And then this side is the right ventricle, and that blood vessel sends blood out to the lungs. So that's the pulmonary artery. Okay. And when we knock out, we can actually have some genetic trickery to specifically eliminate the ETS1 gene just in neural crest cells. And what we see here is a heart defect that does occur in some of your children called double outlet right ventricle. And one of the hallmarks of that is this opening between the 2 ventricles, or a so-called hole in the heart, or interventricular communication, or VSD, ventricular septal defect. But the other thing we see that goes along with this is that the aorta, instead of being aligned with the left ventricle here, it's actually aligned now with the right ventricle. And so that's why we call that double outlet. So both great arteries are aligned with the right ventricle with this hole in the heart. And interestingly, compared to a normal mouse, so what you see here is 3 little things sticking out, protrusions. Those are the 3 leaflets of the aortic valve. And all those blue things that you see there, those are actually the neural crest cells. And we know that neural crest cells have a very important role in the development of the valve. And here, if you look at the mouse now that's lacking the ETS1 gene, you only see 2 of these valve leaflets, and you almost see a complete loss of those blue staining cells. And this is just another way of kind of showing the same thing. Here, the neural crest cells are staining in that pink, and there's a loss of many of those cells when you get rid of the ETS1 gene. Okay, and to give you a little more insights into what's happening, oops, what did I do? So as I mentioned to you, normally like in that cartoon, I showed you how those neural crest cells have to be smart enough to move their way down to that developing heart. So on this top 2 panels is high magnification of a normal developing heart. And what you see here, it looks like a little stream of ants or something. These are individual neural crest cells that are migrating out from that neural tube towards the developing heart. And I think you can appreciate it looks actually very organized. They're streaming right towards the heart. And when we eliminate the ETS1 gene, what you see here is a complete disruption of that normal migration pattern. And it turns out then that loss of ETS1 causes a defect in the ability of these cells to migrate to the area of the heart that's required to do their job to finish developing, in this case, the wall that separates the 2 ventricles and the ability for the great arteries, the aorta and the lung and the pulmonary artery to line up in the appropriate place, if that makes sense. Okay, so loss of ETS1 in the neural crest gives you a ventricular septal defect, and in our mice, a specific type called double outlet right ventricle. Okay, only because they can't get out. And probably a similar story is why a lot of your kids have red hair. Okay, but that's not hypoplastic left heart syndrome. And just like your children, in mice, it really depends upon the genetic background. And you could say each of us, it's a human, has a unique genetic background as well. And so very recently, even since the Pforzheim meeting, we've begun to look a little more carefully at how we might be able to explain those different anatomic subtypes that I told you about. So one possibility, like I mentioned to you, is this excessive inward growth, but that's not really what we most commonly see in the Jacobsen syndrome versions. So another possibility is maybe as these cells start out to form that left ventricle, they're not able to grow outward. So that inability to expand outward might leave you then with this very diminutive slit-like left ventricle. And I'll come back to that idea in just a couple minutes. So before we even discovered the ETS1 gene, it turned out there were some researchers studying this gene in something that maybe I think I've shown to some of you, a fruit fly. And believe it or not, that little thing that flies around and gets in your bowl of fruit in our kitchen, for example, that you want to squash, has actually been a very powerful model for studying heart development. And it turns out, amazingly, that many of the same genes that are involved in the development of the heart in a fruit fly are also involved in the development of our hearts. And I can tell you, kind of going off on a tangent, there's another gene that's related to the function of the ETS1 gene that is clearly, it's called Tin Man, named like the character because when you mutate that gene in the fruit fly, there's no heart. And we have found human patients with mutations in Tin Man, the human version of Tin Man, and based on that knowledge, it's actually led us to do life-saving interventions to prevent them from dropping dead that no one knew about before. So very, very powerful tool. And I always say, think twice before you swat that next fruit fly, because it might actually save your child's life someday. So on the left here is actually a normal embryonic heart of a fruit fly, and you see green, which are the working cells, and then you see these other yellow cells, which actually help to regulate sort of the fruit fly version of blood to come in and out. But very interestingly, this group way back in 2003 showed that if you make a mutation in ETS1, which in the fruit fly they call pointed, look what happens. You get all these excess cells, and it's basically occluding that lumen or chamber. So basically what I'm trying to convince you is this is hypoplastic left heart syndrome in a fruit fly with a little bit of imagination. So that was published back in 2003. So I am very blessed to actually work with the person who discovered that Tin Man gene that I told you about. He works just a mile down the street from where my lab is in San Diego. And so they've been very interested. He's been a fantastic PhD, and he is truly one of the few PhDs that I've worked with that not just for writing grants, but he is truly interested in wanting to know how their research can apply to help our children. And I've got a really moving picture of him meeting that family where we discovered his gene to be mutated that led to the life-saving intervention. It's a really cool story. That's a digression. Anyway, so you lose the pointed gene, the fruit fly version of S1, and you get all these extra cells, suggestive that this is kind of like hypoplastic left heart syndrome. And so more recently, a postdoctoral fellow from Rolf's lab has taken this one step further, and instead of just mutating this gene everywhere, she was able to specifically mutate pointed or S1 in the tissue that actually gives rise to heart cells. And basically, you see the same kind of phenotype. And so that was really nice because that allows us to pinpoint on specific heart cell populations in the fruit fly to help us to learn more about what this gene is doing and what are the pathways that it's regulating. So when Anna Lynn, the postdoc in Rolf's lab, began to get interested in working with us, she actually looked at the literature and found something quite amazing. Fruit fly heart development, even as simple as that little thing is still quite complex. And this just shows you a schematic diagram of some of the genes that are involved in the development of that little tube of cells. And what really blew me away is that there are 3 genes right here that she showed in this. This was just from a review paper from years ago. So Tin Man, that's the NKX 2.5 gene. Pointed, that's ETS1, our gene, and Notch. So guess what? These 3 genes are probably the 3 best known genes that cause hypoplastic left heart syndrome. So in other words, even in the fruit fly, those 3 genes have been conserved across evolution. And taking it one step further, all 3 of these are also regulators like transcription factors. And amazingly, all 3 of these genes converge on what we refer to as a downstream target gene, called even skip, okay, or just abbreviated EVE. It's also a transcription factor. So that might suggest that even skip might be sort of the mediator of these 3 transcription factors that has a critical role in heart development. And so that also led us to ask the question, could there be any patients out there, human patients with hypoplastic left heart syndrome that might have a mutation in even skip? Nobody would have ever thought of that up until us. And sure enough, thanks to my colleagues at the Mayo Clinic who have done a whole genome sequencing on like over 200 patients with hypoplastic left heart syndrome, they found at least 1 patient that absolutely, for sure, has a disease-causing mutation in even skip, or EVX1 is the human name of that gene. And interestingly, just like Jacobsen syndrome children, that child happens to have the aortic and mitral atresia variant. So it really begins to suggest that, don't worry about the details of this, I won't ask you about this one, Jessica. But basically what it says is there are, human fruit fly, you can't think of any 2 organisms further apart from each other, but yet amazingly, there are these conserved pathways now that we think we can justifiably use the advantages of a much simpler system like a fruit fly to study their roles and ultimately begin to hopefully understand how that applies to human heart development and specifically hypoplastic left heart syndrome. So what I want to do is show you some more recent studies that are ongoing, looking at another animal model system that I think I've told you guys about in the past, a frog. So similarly, a frog has most of the same genes that we do. And this story actually started a number of years ago from a collaborator of mine, Marianne Bronner at Caltech up in the LA area. And I just want to show you a few highlights of this slide. So this is in a frog heart. A frog heart only has 1 ventricle. So that's the normal ventricle of a frog heart. And what her then postdoctoral fellow, who is now a junior faculty member at Georgia Tech, was able to do is specifically eliminate the ETS1 gene in the frog, and specifically in that population of cells, like what I told you before, called the mesoderm, that is the progenitor of heart cells. And amazingly, lo and behold, look what you see here. Just focus on this panel. This is what happens, when you take out the ETS1 gene in that population of cells that gives rise to heart cells. You see the pattern here compared to the normal ventricle. You see a thickened wall with what looks like an abundance of heart cells and a much smaller chamber volume. And what's really interesting is, remember I told you about the endocardium? So this green here is a normal frog heart. And what this green is showing you is those normal endocardial cells. And take a look at this. In this same heart, when you knock down ETS1, you have a complete loss of endocardial cells. So what we're seeing here is sort of this theme of when you knock out ETS1, you get a loss of one population of cells going along with this alteration of cardiac myocytes and a very abnormal heart. So this is what a normal tadpole looks like, and it's a little bit hard to appreciate here, but this is what these tadpoles look like when you knock down the ETS1 gene in the cardiac mesoderm. And basically what this is tadpoles dying of congestive heart failure. You can see they're all swollen, and that's just because that heart does not work. So Shuey, who since has now joined, like I said, the faculty at Georgia Tech, is an embryologist by trade. And I think this is just a fantastic example of how it's so great that we have different backgrounds to work together collaboratively. So I want to just kind of show you something more that she's done. So again, this is the normal frog heart. This is just showing what the frog heart looks like when you stain for the cardiac myocytes. So those are red. And what you can appreciate here is that this heart, this is another picture of that same sort of cross section that I showed you of the frog heart, in which we knock down ETS1. And you can see here, it's almost like this ball of cells. It's actually smaller, and it's hard to know at this point if it's actually more cells or if they're just kind of all condensed together. But what Shuey did is, knowing that if you knock this down in that tissue called the cardiac mesoderm, that causes this abnormal heart and loss of endocardial cells, what if you do that same experiment, but now add back normal cardiac mesoderm tissue? And that's what she did. And actually, amazingly, by doing that, you now get back a normal heart. And that is actually a monumental step in the field of congenital heart disease, because imagine if we had this conversation 22 years ago, to think that with early intervention, there may be a window of opportunity to intervene to actually prevent that hypoplastic left heart from developing. Now, obviously, this is a frog. There are a lot of huge steps before we could ever apply this to a human, but at least for me to stand here and have the conversation is, really pretty amazing. Okay, so I think I've kind of reviewed this already. I'll just summarize in the last part here. So these results suggest that ETS1 regulates cardiac cell fate determination, like those endocardial cells that I showed you, and that early restoration of endocardial function may actually be a way to prevent the development of that ventricle. So we wanted to actually take it one step further. It suggests that ETS1 functioning in that layer of cells, the endocardium, is really critical. But to put it to the test more directly, we can actually use some genetic trickery, not to just eliminate ETS1, but to actually just genetically get rid of endocardial cells themselves. And this is what we were able to do a few years ago in the mouse. So this on top is a normal mouse embryo, and these are just different planes showing a normal mouse heart. So this is like the left ventricle of a normal mouse embryonic heart. This is the right ventricle. And if we do our genetic trickery to basically kill off endocardial cells before they have the opportunity to do anything, what you get here is a very, very abnormal looking embryo, growth arrested. And going along with that, these are analogous pictures of that heart, showing that that heart has just completely been arrested. And this is a bit of a stretch, but to me, I think there are some parallels between this heart and that mitral and aortic atresia with the slit-like left ventricle heart. It remains to be seen if we can actually prove that those are the same processes going on. So as sort of a unifying hypothesis, we and others more recently think that the endocardium, at least in some cases, may be the smoking gun, and that anything, whether it's a gene like ETS1 or a genetic ablation of the cells themselves that affects endocardial function, could potentially lead to hypoplastic left heart syndrome. Okay, so really what that translates into actually is that ETS1 has two roles. It's clearly functioning very early in heart development to affect the determination of those cells that I told you about. But I haven't had a chance to tell you actually what it does later on in development. I don't have a lot of time to tell you about that, but I'm going to give one little bit of an example in a moment. We know that normally the endocardium, once it's there, actually sends signals to that ventricle, to those heart cells, to promote normal growth and development. And as I showed you, if you get rid of those hearts, the endocardial cells, you have a very abnormal heart. Those embryos, by the way, cannot survive. So my postdoc Lou Wang in my lab, who's been incredibly talented, did a really cool experiment in the mouse where you can actually knock out ETS1 and compare the pattern of genes that are being expressed in the normal heart compared to what are being expressed in the heart and where you've lost the ETS1 gene. And by doing that, it begins to help you to define what those downstream factors are in this case, the mouse heart. Okay, we already told you a little bit about one potential gene in the fruit fly heart. And so that's what Lou did. And as you can see here, he found a relatively small number of genes that are what we refer to as direct targets, meaning that this transcription factor will bind to the regulatory region of the DNA of these genes and act as that molecular switch. And what's really, really interesting about this is if you look at these genes, so these are 7 or 8 genes that Lou identified that are direct downstream targets of ETS1, at least in the mouse heart. And as an example, ALK1 is a gene that's involved in heart development. We know that. And interestingly, mutations in ALK1 have been found in patients with total anomalous pulmonary venous return. Versus, let's say, ROBO4, mutations in that gene have been found in patients with a bicuspid aortic valve. And we're trying to learn more and more. But at the end of the day, I think this helps us to explain that in any one individual, if you have less ETS1 expressed, in some cases, you might be more sensitive. Your ALK1 gene might be more sensitive to losing half of the amount of ETS1. And in other cases, ROBO4. I'm going to tell you just a very recent development about another gene that may be very, very interesting and relevant to how ETS1 can cause hypoplastic left heart syndrome. And this is a gene called Claudin-5. And this is a paper that was published several years ago, analogous to what I just showed you in the frog for the ETS1 gene. And I don't know how well that's going to show up, but you'll have to bear with me a little bit. This is a normal frog heart from this paper. And these authors, I guarantee you, they don't know what hypoplastic left heart syndrome is, but they were interested in this gene. And what they found is if you knock down the Claudin-5 gene in the frog, like we did with the ETS1 gene, look at this heart. It's a very small hypoplastic ventricle. And so this is very provocative to suggest that the effect of ETS1 on Claudin-5 or maybe other related genes in the mammalian system could be a mechanism that leads to this small ventricle that we see in hypoplastic left heart syndrome. And similarly here, compared to the normal frog tadpoles, you can see when that gene is mutated to give you that very abnormal heart, these are tadpoles that are dying of heart failure. So what does this mean in terms of a human heart? Well, we know when the human heart develops, normally as shown here, you have initially a small ventricle. Actually, this is the developing ventricle right here. But normally in heart development, that ventricle, as the heart begins to take on the 4-chamber formation, expands massively. And that is actually described as a process called ballooning. And what we think could be happening for that slit-like left ventricle is there may be a defect in this ballooning process, so it doesn't allow that ventricle to grow out. And let me just tell you what Claudin-5 does. Claudin-5 encodes a protein that actually helps heart cells to stay attached to each other. It's called a tight junction protein. So imagine if I push out like this, normally if these cells are attached, you can do that, right? If they're loose, it's just going to fall apart. They can't go anywhere. So that's just hypothesis, but based on hard science, it's allowing us to be able to predict some models that we can test in the future. So for example, if you knock down ETS1 in the frog, if we add back Claudin-5, we're going to see if that can actually rescue that phenotype. And if so, that would be really amazing. So ETS1 is expressed in the endocardium and the neural crest, but not in heart cells, the myocytes. In early development, as I said before, it seems to have a very critical role in regulating the determination of heart cell fates. Early intervention may enable rescue of the hypoplastic left heart phenotype by restoring the endocardial function. I also failed to mention that in those rescued hearts, it's early data, but it looks like you do see once again the endocardial cells, so that would fit that model. And interestingly, the specific cause probably determines whether or not the cardiac myocytes, that's pumping cells, are actually intrinsically normal or maybe intrinsically abnormal. So in Jacobsen syndrome, remember what I told you, S1 is not actually expressed in cardiac myocytes. It's due to the abnormal function of the endocardial cells that then impairs those cardiac myocytes. But there's a very interesting clinical implication of that. The clinical implication of that is that initially those cardiac myocytes are intrinsically normal, which means there may be opportunities, if one could intervene early enough, to rescue those cardiac myocytes in a way that might restore the normal development of the heart. So I'm going to go off on a little bit of a tangent here for the last few minutes. How are we doing for time, by the way? I'm losing track. How much do I have? I was supposed to be done 10 minutes ago. All right, we're doing okay. Okay. But before I do that, just to summarize, there's no one ideal model system. And so we've combined, not just myself, but with some incredible collaborators, human studies, okay, keep me on target here, past studies and ongoing studies in fruit flies, zebrafish, frogs, and mice. So as I just alluded to, one interesting question then is it possible for Jacobsen syndrome and maybe other patients, subsets of patients with hypoplastic left heart syndrome, are those cardiac myocytes salvageable? There may be other cases where, the die is cast and you can't do anything to overcome that. And actually, interestingly, consistent with this hypothesis, recently there were some investigators from Toronto who showed that relatively late in heart development, if you just block off the mitral valve, you get hypoplastic left heart syndrome. And those are genetically normal mice. So that tells you that, at least if you believe that model, that there can be intrinsically normal cardiac myocytes. And again, that has some really important clinical implications. And I want to just show you something really cool that a company that I've started to establish a collaboration with will allow us to, in a formal way, test this. This is a company that's based out of Taiwan called NovoHeart. It's referred to as an academic spinoff. So it's started by MDs and PhD cardiologists. And in this case, what they're able to do is actually take either a normal patient or a patient with hypoplastic left heart syndrome. Some of you may remember years ago in San Diego, we got skin biopsies. Yes, you're scratching your. So there is now the technology where we can take skin cells and through some trickery, genetically reprogram them to become heart cells. And what these guys at this company are able to do is recreate, in this case here, what you see is actually what they call a heart in a jar. And if you look closely, this heart in a jar is actually beating. And when I talked to these guys, I said, if you had told me 10 years ago that you could take heart cells derived from human skin cells and create an actually beating ventricle, I'd say you don't have a freaking chance. But this is what they've been able to do. Oops. And what's really amazing is we can use this to characterize function of a ventricle and correlate that with clinical outcomes. Because as we know, some kids with hypoplastic left heart syndrome, by the time they're school age, develop heart failure of that right ventricle. And others, fortunately, do really, really well later, living much longer. And so if we could determine that ahead of time, imagine how helpful that would be to stratify which patients maybe we need to list earlier for transplant, which patients maybe need to be started on heart medicines earlier on, like enalapril and other heart failure medications. And even it might help us eventually to determine which patients could be potentially candidates for procedures that could restore the function of that left ventricle and the growth. So basically, I just kind of got ahead of myself, and this is what I'm saying here. We can also actually use this system to identify new drugs that can hopefully improve function specifically on heart cells from a patient that we know, let's say, develops early right heart failure. And it's also actually amenable to studying other types of heart disease as well. All right, in my last few minutes, this has nothing to do with Jacobsen syndrome, but I just want to give you a little bit of a kind of a cool show and tell about some of the other things we're able to do. And it actually comes back to the story I told you before about cardiac neural crest cells. So one of my colleagues who does fetal echocardiograms told me recently about a fetus that they were referred to from an obstetrician in San Diego who was found to have a very, very abnormal fetal echocardiogram. And what they actually found that in this 22-week fetus, just from the routine screening ultrasound, was that this fetus had an extremely rare heart defect in which that aortic valve that I showed you pictures of before was completely absent. And this is a picture, it's actually an incredibly beautiful picture, if you're a cardiology nerd like me, of an echocardiogram of that fetus's heart. And this is the left ventricle and this is the right ventricle. In this case, the left ventricle is massively dilated. This is the chest wall here and here. So basically, this fetus's left ventricle was completely blown up, taking up all the space in the heart, in the chest. And the reason for that is there was no ability for this left ventricle to pump blood out to the body because there was no valve. Remember what valves do, right? They prevent blood from leaking backward, just like valves in a car engine. So in this case, and don't worry about this, this is just another way to assess flow, but that red flow that I'm pointing to basically shows that all that blood goes backwards. Okay, so histologically, this baby was basically dying because that heart function was already, severely impaired, and they actually terminated the pregnancy literally just a couple of days later. But our pathologists were able to do a systematic analysis of that heart. This is just a section through the aorta where you would normally see the heart valve. And what this blue is just scar tissue. So this confirms the echocardiographic results that this was a completely absent aortic valve. Only one other case reported in the literature. So when my colleague told me about this, I said, God, wouldn't that be cool if we could get that fetus's DNA and see if we can take advantage of what we have in San Diego, whole genome sequencing. And that's what we were able to do. So this baby was actually born about 2 years ago. They did an initial kind of an array CGH and nothing came back. So I was able to go on a wild goose chase and track down the lab that actually did that initial array CGH analysis. And they still had some DNA sitting there in a tube, in a freezer somewhere. And they sent that to our Genomics Institute at Rady, which is one of the few in the country that does in-house whole genome sequencing. And literally within a few days, I was out running around the track on a Saturday at my son's school, and I got a text from the director of the Genomics Institute, and he said, hey, Paul, we found something really interesting. And what they found was this fetus was found to have a mutation in a gene called APC, which don't worry about what it does, but historically, it's actually a gene that causes something called adenomatous polyposis coli, which is having a bunch of polyps in the intestine that can predispose to colon cancer early in young people. However, that's not relevant. What is relevant is if you dig through the literature, it turns out, remember I told you the story about neural crest cells and the development of the aortic valve? Well, it turns out this gene actually plays a critical role in neural crest cell survival. And so there's something called an apoptosis or programmed cell death or cell suicide. So this gene normally puts the brakes on that pathway. So it's called a tumor suppressor gene. And it has been shown in mice that if you mutate this gene, like what we found in this kid, you lose that tumor suppressor function. And lo and behold, these neural crest cells die. So we can't quite prove it, but it speaks to the very likely possibility that this mutation resulted in early programmed cell death that killed off these neural crest cells that are required for that aortic valve to develop. And so it's just a really cool example of kind of the resources that we have right in-house in San Diego to learn new things. Okay, before I stop, I want to acknowledge not just all of you guys as I did the other day with your incredible support, because without that, I would have just shown you one slide that is blank. So instead, I hopefully showed you that your support has gone to good use. So Aiden is actually an undergraduate at UCSD who's working in the lab when he has time, hoping to go to medical school He still wants to be a pediatric cardiologist. This is Lizu Lin, who is a senior staff scientist who did all that beautiful work on the neural crest studies. I didn't get a chance to show you all of that. That paper just got submitted, and it looks like preliminarily it has been accepted to a really nice journal called Human Molecular Genetics. Unfortunately, because I told you that my single biggest donor passed away a year and a half ago, I had to let her go recently because she's so good I can't afford her. This is Anna Lynn Schroeder, who has done the fruit fly experiments that I told you about with my colleague Rolf Bodmer just down the street in La Jolla. This is Lou Wang. Lou is, without a doubt, he is the most amazing postdoctoral fellow that I ever had in my 20-something years. He has done all the experiments that I showed you in a very brief way, actually, about this whole story of how ETS1 functions in those cells called the endocardial cells. He came here from China. His English vocabulary, I think, is actually better than mine. I mean, this guy is just amazing. And he's nearing the end of his tenure as a postdoctoral fellow, but I'm going to do everything I can to keep this guy because he is just absolutely amazing. And then this is Antonella, who is a now junior researcher. She actually did some really cool experiments that I didn't have time to tell you about. Similarly, where we can actually take human patients with heart defects and reprogram them to become neural crest cells. And basically what she was able to show in human neural crest cells from patients is that they have the same kind of migration defect that I showed you in the mouse. And she actually allowed us to begin to understand with those human neural crest cells why you might have a heart defect in one case and not in another. And the reason being, you know, when you have a deletion in chromosome 11, you have one normal copy of the gene, right? Supposedly normal. What she found is that there are massive differences in whether or not that one normal copy of the gene is expressed and functioning. And so we think one potential possibility to explain why half of your kids have a heart defect and half of them don't is whether or not that one copy is working well. So that's really cool insights. That's just a few of the people from my lab most recently, but as I've alluded to, there are a number of people both locally in San Diego and literally from around the world that really truly make this, I think, a superstar, all-star team, you know, really incorporating a multidisciplinary approach. And I think really at the end of the day, you really have to have PhDs, MDs, and everyone in between working together, because just this last week we had Wednesday, actually, I skipped out of here to have my lab meeting, and it was just a great example of how you get perspectives from different areas that come together to give us insights that truly none of us would have come up with on our own. This is another version of what I showed you the other day, a lot of different sources of funding. And like I said, without this and without you guys, it would just be a blank slide. So that's it. Thank you. Okay. Oh, you know what? Did I mute? I forgot to mute it or unmute it. Did they not hear anything I just said for the last hour?
No.
Okay.
First question is here.
Ah, there we go, up there. Okay, I read that one of the symptoms, no, crowded toes. I'm not gonna answer about crowded toes here. Okay, so the question, I'll just read this. This is from Eleanor Block to everyone, so I think she's okay for me to share this. She says, since Dr. Grossfeld is in California and Jacobsen syndrome is relatively rare, can he please suggest how we can communicate to our local cardiologist the types of tests, procedures, protocols that he or she might do for our child with Jacobsen syndrome? Well, the best way that I can answer that is a few ways. If you go to the 11QUSA.org website, you will find 150 papers that talk all about different aspects of Jacobsen syndrome, including our papers that give specific recommendations. I'm happy to talk to you further offline about what that should be, but definitely anybody with the new diagnosis of Jacobsen syndrome deserves a comprehensive baseline cardiac evaluation, which should include an echocardiogram. Yep.
Any way to regenerate the loss of neural crest cells?
Repeat question.
The question is there any way to regenerate loss of neural crest cells? Not that I'm aware of clinically. However, there's something that is kind of related to that. For all of you that have been pregnant, do you remember that you took folate? So do you know why people take folate when they're pregnant? Any idea? It's, yeah, it among other things, it prevents neural tube defects, and the mechanism that's thought to be underlying how it works is that folate actually preserves neural crest cell proliferation. So that's what, you can't go back, I don't think, when the horse is out of the barn, so to speak, but maybe that's the closest to answering your question. Yeah.
I can't imagine that there are too many people growing hearts in jars, but I did hear on NPR, and I mentioned this to you the other day, the story of how they did that, and I'm assuming it's what you spoke of, but they were talking about how they created it with just the cellular level, and it grew, and then began eating on its own, or maybe with some intervention. So I guess is there any? You're talking about early intervention in utero of potentially solving the dilemma of hypoplastic left heart. Is there any future, might be science fiction now, but down the road of being able to grow with one's own cells, one's own heart, and replace it later? Have a heart transplant with your own heart.
Well, one of the problems is if you're born with hypoplastic left heart syndrome and you assume it's a genetic cause, if you take your own cells to create a new heart, you might predict that you're going to be recreating the same problem, right?
With the intervention.
So if you do an intervention, it does sound like science fiction right now, but I can tell you that this is not the only example of what people are what's called creating organoids, whether it's a heart. It's a little more controversial when people are creating brain organoids. That's pretty dicey, but so people are on that trail. It's very, very early, but stay tuned. The other thing where I think this might hold some promise actually is it actually fell out of favor a number of years ago, but it's regained a lot of publicity lately, and that is xenotransplants, and specifically from pigs. So, every time you have your side of ham or a piece of bacon, think about how that heart just gets thrown out. But what people are doing now is genetically re-engineering pigs so that those hearts are going to be more compatible for transplanting. One can question the ethics of that, but if millions of pigs are slaughtered every year so you can have your morning bacon, why not take a pig with a genetically engineered heart that could be actually an unlimited supply? And so, recently, if I'm remembering correctly, there was a pig heart that was put into basically a brain dead patient just to test the hypothesis if it could function. I think, was it a brain dead person? It was also done with kidneys, but anyway, stay tuned, because that I think might be a little more, it sounds like science fiction now. I think the whole organoid field holds a lot of promise, but still with a lot of challenges. But I could much more realistically envision a pig heart genetically engineered so it will be compatible and not be rejected. That could potentially be like an unlimited source, as unlimited as your breakfast bacon is. So yeah, go ahead, Beth.
Earlier today, Dr. Harris had mentioned about how the immune system could kind of go downhill after a while. Is there anything or any research that shows that something that similar could happen with heart issues? So if somebody like you said, ASD and VSD, they closed up on their own, is that something that we need to, every so often we follow up, so far it's been good, but
Right.
is that something that there's any research?
So it's a great question. This is one of the reasons why, it's not just like one and done. When someone has a relatively simple heart surgery, like closing a hole, you would think in theory, hole is closed, you're done, I never need to see you again. We still follow those patients every year or two just to keep an eye on things. I am not aware of anybody specifically with Jacobsen syndrome who developed, like late onset heart failure or cardiomyopathy. So that's the best answer I can give to you.
Early on when you were talking about, well, some years back when you were talking about the ATS1 and the way that it was split, And there was some talk or association with how it was affecting the neural developmental delays. Is there any new information or, new found information or confirmation that that is a thing?
Right, not really. I mean, I've got to keep my limited time and resources focused on the heart story. So we do know that ETS1 is expressed, I think, in the developing brain. Lizu from my lab thought maybe there were some behavioral patterns that were different in the ETS1 knockout mice. So I wouldn't be surprised if it could potentially do something, but again, from one person to the next, it's very variable. So, but yeah, that's a little beyond my resources to pursue that part of ETS1. We do know, just as an aside, that ETS1 does also cause structural kidney defects that occur in about 10% of your children. So, yeah. Who else? Cool.
Nope.
That's it?
That's it.